Hey everybody, my name is Eric Prileson and I'm a second year master's student at Case Western Reserve University in Cleveland, Ohio. And in this forum, we're talking about evolution over contemporary timescales. But I find it really fascinating to think about how evolution could occur at small spatial scales or microgeographic spatial scales. And thinking about this, it's interesting to think about how you could use the city or an urban habitat in order to explore that. And that's what this study entails. So natural selection can occur when there's a heterogeneous landscape and possibly result in evolutionary divergence within populations. And despite the influence of gene flow or dispersal across small spatial scales, even when populations are relatively close together, it's possible that divergence can occur at that microgeographic or small spatial scale level. For instance, if we can imagine these two fruit fly populations that exist on two different types of host fruit trees, and there is selection acting on the host tree fruit for some particular traits such as nutrition or food preference, you could actually have divergence despite the fly's ability to disperse between them, where selection is acting and that is a trait that's influencing fitness. How exactly could this work? So how exactly does this divergence work? Well, if there's a high strength of selection or possible barriers to gene flow or dispersal, you could result in microgeographic divergence. And this is depicted in this diagram on the right side, where hypothetical populations in green, where there's no microgeographic divergence at small distances between each other, if there's a high strength of selection, perhaps for those fruit fly traits, if it's higher than expected, that can move the population towards that orange area, which is outlining a possible microgeographic divergence, right? So if there's a high strength of selection, or if there's barriers to gene flow, then this could result in a possible microgeographic divergence. However, it's unclear when we are expected to find microgeographic divergence versus other possible outcomes, such as phenotypic plasticity or possibly maladaptation. Now, urban habitats or the city provides a great design for studying evolution at small scales because cities are naturally highly fragmented and variable due to the urban road network and the mosaic-like pattern of habitats and patchwork that makes up the city environment. You can get a good glimpse of this if we look at the city of Cleveland here overhead in the satellite image, lots of patchy environments and habitats that could lead to structured populations between them as long as the same species and populations within that same species exist among these patchy habitats. And so we need to find a species to explore this idea that fits that mold that exists within these structured mosaic-like habitats. And so we decided to use a collembolan, a springtail, in this case the species Orchicella viosa, depicted here, which is a very distinctive springtail species. But interestingly, this species is dispersal limited. It's quite small, and it's obviously unable to disperse beyond or unlikely to disperse beyond areas that are fragmented by the urban road network. And so it's hypothesized that these populations within the city are highly structured. So they make a good system for exploring our question of interest of microgeographic divergence. And so our goal with this design, it was to test possible evolutionary divergence in two environmental variables at small spatial scales. We wanted to look at divergence in responses to differences in variation in temperature and in road salt pollution, two important factors that are a part of urban life in the urban habitat. And so we selected sites to sample our populations of Orchicella viosa from across a range of soil salinity and different temperatures. In order to select sites across a range of different soil salinities, we use traffic data, assuming that higher traffic levels scale with the amount of road salt applied and therefore the amount of road salt pressure. And so this is locations that have traffic data from the Ohio Department of Transportation. And so this gave us a very large list of potential locations to search from. We narrowed this list down a bit by using Google Street View to only find locations where we would expect to find our species of interest, areas that would have leaf litter, such as this location, for example. And this narrowed it down to about 110 sites across the city of Cleveland, shown here. And we also wanted to select from sites that had variation in temperature. And we used impervious surface area and variation in impervious surface area as a metric of temperature, since impervious surface is or is a causal influence of the urban heat island or differences in temperature of the urban area, right? And so each of these locations we scouted to see if we could find our species of interest, and we used Bicycle to scout and locate and find our species of interest. And ultimately, we were able to find Orchicella viosa at 25 locations of these locations across the city of Cleveland, shown here in these yellow pins. The differences in the color of yellow indicates the scale of soil salinity, with lighter color yellow indicating higher soil salinity. And the map here depicting impervious surface, darker colors of blue indicate higher impervious surface, so indicating warmer temperatures, versus lighter blue or white indicating cooler temperatures. So this is how we selected our sites and sampled our populations. So at each location, we accessed, as I mentioned, by a bicycle, and we collected 30 to 60 individual springtails per site. We took some site level measures, including temperature, and we used some site level soil conductivity measures to see that particular measure and use that particular measure, since salinity did not scale with traffic as we had expected. And we also wanted to get some longer term temperature measures to see how well impervious surface scaled with temperature within the urban heat island itself, to see if that variation was consistent with ISA and temperature. And there's my high school student helping out with our collections knee deep into finding springtails in the leaf litter. And so we brought we wanted to find if there was variation, to possibly see if there's phenotypically plastic responses to temperature variation or road salt pressures, or potentially evolved differences. So we tested both field caught individuals of springtails as well as laboratory reared individuals under common garden conditions. And so we brought our springtails into the lab and first tested heat tolerance in the field caught generation of springtails, the F sub 0 generation. And then we also tested heat tolerance in the F sub 1 generation, or the laboratory reared generation, in order to minimize those environmental effects. And so we can attribute any remaining variation to genetic divergence. In addition to heat tolerance, we tested a subsequent generation, laboratory reared generation, tested their salinity tolerance of laboratory reared springtails, again, to see if there's that potential evolutionary divergence of that particular trait. All right, and so to test heat tolerance, we assayed using this critical thermal maximum, or CT max, and this is a dynamic ramping protocol, increasing the temperature slowly over time at a half a degree Celsius per minute until the springtail loses motor function. In addition, and that temperature is deemed the CT max, or critical thermal maximum. For salinity tolerance, the other physiological trait of interest, we split the springtails into three different soil substrate measures, with three different soil or three different saltwater concentrations added to that soil substrate, and then tested their survival over a 12-week period. So what did we find? I'm going to start with the salinity data first, just to showcase that. So with our salinity tolerance, we had expected that springtails from higher road salt pressure environments, so sites that had higher soil salinity levels, to have greater survival than those springtails from lower soil salinity sites. And we also expected the same pattern from springtails from higher soil salinity to have higher survival under higher saltwater concentration pressure. But interestingly, we don't see any difference across our origin site soil salinity in our results. And also, we don't see any difference across the different soil salinity treatments. So we don't see any evidence for divergence in salinity tolerance with the springtails within the city of Cleveland. Okay, what about heat tolerance? Well, first, let's showcase temperature differences. Is there variation itself within the city at different sites with different ISAs? And the answer, with those longer term temperature loggers that we use, the answer is yes. Higher impervious surface sites tend to be warmer, as we can see in this graphic, up to a point. Okay, so in this case, we might expect springtails from populations from higher impervious surface or higher ISA to express higher heat tolerance. And so let's look at our data from the field caught generation of springtails. So just briefly walking you through this figure here, we have a contour plot where impervious surface is on the x-axis and salinity or soil conductivity is on the y-axis. And each of the contour lines is depicting heat tolerance, where warmer colors or yellower colors are depicting higher CT max and cooler colors lower CT max. And interestingly, we have a significant interaction of impervious surface and soil salinity that are explaining the variation in heat tolerance, which is pretty interesting. So this correlates with our expectations, where we had expected populations of springtails from higher ISA locations to express higher heat tolerance, which seems to be the case here, but it's dependent on a certain level or threshold of soil conductivity. Shown here, we can see that peak of heat tolerance on the right side of higher ISA, but only for those populations that have a certain threshold or certain level of soil salinity at that location. So we have that significant interaction explaining the field caught generation heat tolerance, which is pretty interesting. Okay, so does this pattern of an interaction, is this consistent? Do we see the same pattern in our laboratory reared, which would indicate an evolutionary divergence in populations from different impervious surface locations? Well, in our laboratory rear generation, we actually do not see that pattern. There's no interaction. So when we model this relationship of impervious surface on heat tolerance, we actually see a negative relationship where populations from a higher ISA location express a lower CT max or heat tolerance, which was unexpected. So this is interesting because we have a difference between our field caught generation springtails in heat tolerance and the F1 generation of springtails and their heat tolerance. Right. So with these differences, right, we have evidence for both plasticity, that the differences are expressed between the F0 and the F1 generation, and also an evolved difference in CT max, where in the F1 generation, the low ISA populations are expressing a higher heat tolerance compared to the field caught generation. Compared to high ISA location springtails. And so this is interesting because heat tolerance can be influenced by and vary across a range of developmental temperatures, right? We can consider this a reaction norm. And reaction norms themselves can undergo evolutionary divergence. And so we expect that there are two potential ways that the reaction norms have evolved. And we can't say for sure with our results here because we only reared springtails under one developmental temperature, the 20 °C temperature. So there are two hypothetical ways that we expect these reaction norms to go. In scenario A, we could expect, or we could see, that populations from a lower impervious surface location continue to express higher CT max across a range of developmental temperatures, right? And this parallel reaction norm in comparison to high ISA springtail populations could be evidence for maladaptation for high ISA populations, right? They're exhibiting lower performance of heat tolerance at all temperatures. Alternatively, in scenario B, we could see crossing reaction norms where populations of springtails from high ISA locations express higher heat tolerance as rearing temperature or developmental temperature increases, right? And so this would be evidence for a genotype by environment interaction where heat tolerance is dependent on both the developmental temperature and the population type. So we obviously couldn't determine that from our experiment and our results here because of our design, but future investigations could set up common garden tests across multiple rearing regimes, multiple temperature rearing regimes to disentangle those possible outcomes. But complicating this is that interaction of soil salinity we found in the field caught generation. So will rearing under different salinity regimes in addition to different temperature regimes reveal those continued interactions? That's an open question which would be really fascinating to investigate. So overall, while there's an evidence for microgeographic divergence in heat tolerance, the lack of evidence in salinity tolerance, which is interesting, it actually could be a function of the variability of the road salt pressure, right? Winters are not always the same, and so that could vary year to year or seasonally with different levels of precipitation, causing different or fluctuating levels of road salt pressure to populations of soil-dwelling critters like our springtails. And so a fluctuating pressure at small spatial scales, these microgeographic scales, could favor more of a generalist response for certain pressures. And so this suggests that outcomes at microgeographic scales could be context dependent, especially for those pressures that are not temporally stable. And so with that, I'd like to thank my advisor, Dr. Ryan Martin, collaborators, Dr. Sarah Diamond, Dr. Gideon Deem, and my high school student, Matthew McCormick, funding from the CWRU Oakle Bay Fund. And thank you so much. I'd happy to take any questions.